Hello, my project was on intermittent fasting's effect on brain function and cognitive well-being. First, I want to talk about what is intermittent fasting. It's a type of diet that restricts the hours of which someone is allowed to eat. There are two main approaches to this. First, the 16-by-8 approach, which is fasting for 16 hours, then eating for the next 8 hours. As for the 5-by-2 approach, which is less known, it's eating normally for 5 days a week, then only eating 500 to 600 calories for 2 consecutive days. This type of dieting causes ketones to form during fasting periods. The body does not have carbohydrates to use, so it attacks fat, causing ketones to be the primary source of energy for the body, and that does include the brain. As for the studies I looked at, the first one was intermittent fasting protects against the deterioration of cognitive function, energy metabolism, and dyslipidemia in Alzheimer's disease-induced estrogen-deficient rats. As for the methods and procedures, there was a group of female rats that were put into 4 groups. First was the Alzheimer's disorder with normal eating diet. Then there was the Alzheimer's disease intermittent fasting rats. Then there was the non-Alzheimer's disease normal dieting. And lastly, the non-Alzheimer's disease intermittent fasting. What I mean by normal dieting in these groups is they were given free rein on food without any type of diet. Now, the first week of the study allowed the groups to get acclimated to their surroundings. In the second and third weeks, the rats were given a high-fat diet and given their specific B-amyloid, which is related to whether they were given a higher chance for Alzheimer's disease or not. In the 4th week, the trial started and continued for 4 weeks. The intermittent fasting group was given 3 hours a day to eat, and the non-fasting group had free range to eat food all day. After 8 weeks, the researchers used behavioral, biochemical, and physiological tests to see the effects of the study. As for the results, the Alzheimer's disease rats showed increased B-amyloid deposition in the hippocampus, a hallmark of Alzheimer's pathology. The rats in the intermittent fasting group, however, showed less B-amyloid deposition compared to the no food limitation group. The non-Alzheimer's disease rats, which were the control, had minimum B-amyloid deposition, and the fasting regimen, intermittent fasting in this case, did not alter this minimal deposition. This confirms that the effect of intermittent fasting on B-amyloid accumulation is specific to the Alzheimer's model. Now, as for the Krebs phosphorylation or CAMP response, a limited binding protein, a crucial protein involved in memory and learning, was reduced by B-amyloid infusions in the hippocampus. This indicates impaired memory function. In the intermittent fasting groups, it protected against this reduction in Krebs phosphorylation in the Alzheimer's disease rats, suggesting that intermittent fasting could support memory-related signaling pathways even in the presence of Alzheimer's pathology. In the passive avoidant test, the Alzheimer's disease rats showed a decreased latency in entering the dark chamber, indicating memory impairment in the form of short-term memory deficiencies. Intermittent fasting particularly prevented this memory impairment, as the Alzheimer's disease intermittent fasting had longer latency time compared to the Alzheimer's disease normal eating group. This shows that intermittent fasting helps preserve short-term memory in Alzheimer's disease rats, likely through its effect on neuroprotective pathways like Krebs and insulin signaling. As for the test I just discussed, it just was looking at the rat's short-term memory. Now, as for autophagy and intermittent fasting, this is really important because it was discussed a lot in the previous study as to why intermittent fasting was able to help with the B-amyloid reduction. Now, as for autophagy, it's a cellular cleaning process where the body breaks down and recycles damaged or functional protein, organelles, and other cellular components. This process helps maintain cellular health, clear out waste, and even promote tissue repair. It is crucial for longevity, immune function, and overall cellular function. Intermittent fasting reduces B-amyloid depositions in the hippocampus and protected against memory impairment, which is consistent with the idea that autophagy induced by fasting helps maintain cellular health in the brain. Now, as for my second study, it was brain responses to intermittent fasting and the healthy living diet in older adults. As for the methods and procedures, 40 adults with obesity and insulin resistance with an average age of 63 were put into two groups. First was the intermittent fasting group, which followed the 5-2 rules of intermittent fasting, consuming only 480 calories for 2 days, then a regular diet the other 5 days of the week. As for the healthy living group, they ate the USDA-approved healthy diet every day of the week, which includes fruits, vegetables, lean proteins, whole grains, and low-fat dairy. This study lasted 8 weeks. As for the tests they did during the study, there was an extracellular vesicle analysis. The researchers isolated these vesicles to study insulin-resistant markers and other brain health biomarkers. They also did a brain age, which is a measurement of the biological age of the brain based on structural MRI data. This test assesses how much older the brain appears compared to its actual chronological age. They also did an MRI, which is an imaging of the brain. They were used in conjunction with the brain age that I just talked about. As for the cognitive function test, these tests assess abilities like planning, decision-making, goal achievement, and working memory. There was also measurements of metabolic health, which included weight and BMI, blood lipids, insulin resistance, and there was also a glucose concentration in the brain, which researchers assessed the glucose concentration in the brain as well as the overall glucose metabolism. There was also neurofilament protein levels, which looks at the levels of neurofilament light chains, NFL, a protein found in neurons, to measure neuron damage or neurodegeneration. I also added a brain age imaging on the screen. As for the results, both intermittent fasting and healthy USDA diet showed benefits for reducing insulin resistance, improving cognition, and enhancing brain health biomarkers. The intermittent fasting group, however, demonstrated a greater cognitive improvements, especially in memory and executive function. It also followed more favorable effects on brain health biomarkers like neurofilament proteins. The intermittent fasting also caused more weight loss than the healthy living diet group. Although these diets show improvement in both brain and metabolic health, biomarkers of Alzheimer's disease did not change with either diet. This is interesting considering the last study I talked about says the opposite. I think that is related to the small studies that have been performed on humans, and the other study was done on rats. I also added a picture of the multiple groups that they had and the results that formed. Now, as for my last study, it was calorie restriction improves memory in elderly humans. As for the methods and procedures, the studies used 50 healthy elderly individuals who were normal to overweight and were put into 3 groups. First, there was a calorie restriction group, which decreased calorie intake by 30%. There was also an increased unsaturated fatty acids groups, which there was a 20% increase in the intake of unsaturated fatty acids while still consuming the same amount of fat as they normally would. There was also a control group with no dietary changes. The study lasted 3 months. Participants were assessed on memory performance, various physiological and metabolic parameters, and insulin and inflammation markers at baseline and after the intervention. As for the results, participants in the calorie restriction group showed a significant improvement in verbal memory by 20%. The improvement was correlated with reduction in fasting plasma insulin and C-reactive proteins, which CPR is an inflammation marker. This suggests that better memory performance was linked to improvement in metabolic health, particularly insulin sensitivity and lower inflammation. There was no significant changes in memory performance for the unsaturated fatty acid groups. This indicates that a diet rich in unsaturated fatty acids did not have a measurable impact on cognitive function in this study, at least over the short term. The control group saw no changes, and within the study, there were no changes in the brain-derived neurotrophic factors, suggesting that while metabolic changes might have played a role in improved cognition, the changes in neurotrophic signaling pathways did not, and that is according to this study at least. As for the social relevance and what is missing within these studies, as people get older, normal cognitive functions such as memory decline, but with the studies I just described, intermittent fasting can be a way to improve memory and delay the onset of cognitive decline. Research on intermittent fasting and brain cognition is still limited, and I was only able to find one study directly relating intermittent fasting to cognitive improvements in humans. There is a lot of research out there on intermittent fasting and cognitive health, but most of it is related to animals or rodent-based. These are great preliminary research studies, and with the rise of popularity of intermittent fasting, I can see more coming in the future. I also wanted to point out that the last study I discussed does not directly talk about intermittent fasting, but calorie restriction. Intermittent fasting is a form of calorie restriction, and the two diets have similar body responses. I wanted another human-based study to be discussed, and from this research, and from research in general, I felt the two studies were close enough to compare. As for the overall for all these studies, there seemed to be positive relevance with intermittent fasting and cognitive and brain function, so I can see more studies to come that just show the improvement within human studies. As for now, we can only look at the animal and rodent-based and the few human studies we have. Here's my works cited, and thank you so much.